Integrative therapies hold some promise for people with idiopathic pulmonary fibrosis. Although most of the evidence at this time comes from lab and animal studies, based on the current understanding of the underlying mechanism in idiopathic pulmonary fibrosis, integrative therapies that target restoring the oxidant-antioxidant balance and preventing cellular microinjury, as well as those with anti-fibrotic and anti-senescence properties, are of interest. N-acetylcysteine is a form of the sulfur-containing amino acid cysteine. N-acetylcysteine reduces oxidative stress, and it's been used as a powerful antitoxin. It's been shown to break down mucus secretions, making it easier to clear them, and it's used for this reason to treat pulmonary fibrosis, associated with genetic diseases like cystic fibrosis. N-acetylcysteine has been shown to reduce the oxidative stress and the inflammation, and to inhibit fibrosis in the lab and animal models of idiopathic pulmonary fibrosis. Studies in idiopathic pulmonary fibrosis patients have yielded conflicting results. In one randomized control trial in idiopathic pulmonary fibrosis patients taking prednisone and azathioprine, the addition of 600 mg of N-acetylcysteine 3 times a day for 1 year led to slower disease progression and better preservation of lung function, but it didn't statistically alter the mortality. In another clinical trial that included 155 subjects with idiopathic pulmonary fibrosis, about half of them were treated with prednisone, atropine, and N-acetylcysteine, and the combination was associated with increased rates of adverse events and mortality when compared to placebo. The same researchers found the treatment with N-acetylcysteine alone had no impact on the disease progression relative to the placebo, and although the N-acetylcysteine was associated with better walking capacity and quality of life, these differences were not statistically significant. Genetic variation can help to explain these inconsistent findings. Researchers discovered that variants with a specific gene can predict the response to N-acetylcysteine. When participants from clinical trials were grouped according to the gene variant, those with one variant fared worse than the placebo when treated with N-acetylcysteine. Those with another genetic variant had no response to the N-acetylcysteine, and those with a third variant showed significant improvement when treated with N-acetylcysteine. It's hoped to give a better understanding of the gene variation drug therapy interaction, and that this can help individualize the treatment in the future. Niacin, or vitamin B3, is the main precursor for nicotinamide adenine dinucleotide, or NAD+, an essential cofactor for cellular activities related to metabolism and energy production. Levels of NAD+ decrease with age, and are associated with metabolic dysfunction, while raising cellular NAD+ levels has been shown to increase the activation of certain anti-aging proteins. Because idiopathic pulmonary fibrosis has a close relationship with aging, it's thought raising the NAD+ can be beneficial. Findings from several studies using animal models of idiopathic pulmonary fibrosis indicate that niacin administration might prevent the depletion of the NAD+ in the lungs and reduce fibrosis. Other research in animal lung cells show that combining the niacin with taurine might have a unique benefit. Taurine is a conditionally essential amino acid, and it's known to neutralize certain toxins and protect against oxidative tissue damage. Evidence from preclinical research suggests that taurine and niacin can inhibit the activity of pro-fibrotic inflammatory molecules. It can suppress free radical production in the lungs and ultimately reduce collagen deposition in the lungs. Other NAD+ precursors like nicotinamide riboside have been studied in the context of pulmonary fibrosis. However, some preliminary evidence suggests nicotinamide riboside ameliorated the liver fibrosis in a mouse model of non-alcoholic fatty liver disease. Alpha-linolenic acid is an essential omega-3 fatty acid, and it's found in flaxseed and some other plants. It's the precursor to EPA and DHA, which can also be obtained from fish oil and has been reported to have numerous health benefits related mainly to their anti-inflammatory effect. In one study, replacing all the dietary fat with fish oil protected the rats against an agent that's used to induce an experimental idiopathic pulmonary fibrosis-like condition. A similar protective effect was seen in a study where mice were fed a standard lab food plus flaxseed oil. In another study, mice who were treated with DHA prior to exposure to the same fibrotic agent had lower levels of inflammatory molecules in the lungs and the alveolar fluid, less lung fibrosis, less weight loss, and lower mortality. In two other studies, treatment of mice already exhibiting idiopathic pulmonary fibrosis-like illness with resolvin D1 or protectin DX, lipid molecules derived from the metabolism of DHA reduced the inflammation and the fibrosis, and it improved pulmonary function. Dehydroepiandrosterone, or DHEA, is an androgen hormone and is produced mainly by the adrenal glands. Levels of DHA and its sulfated form, DHEA sulfate, decrease as we age. Low levels are associated with chronic diseases and frailty, and higher levels have been correlated with health and well-being. Blood levels of DHEA sulfate have been noted to be significantly lower in some individuals with idiopathic pulmonary fibrosis when compared to healthy controls. In addition, DHEA inhibited lung fibroblast proliferation and migration in the lab research, and it indicates that it has a potential usefulness in idiopathic pulmonary fibrosis treatment. Clinical trials have not yet investigated if the DHEA replacement therapy benefits the idiopathic pulmonary fibrosis patients. Vitamin D has many beneficial immune supportive and anti-inflammatory effects. Chronic vitamin D deficiency has been noted in lab animals to increase the production of extracellular matrix components like collagen and to promote fibrosis in the lung tissue. One interesting study examined the influence of the season on mortality related to pulmonary fibrosis, including idiopathic pulmonary fibrosis. The study found that compared with the summer months, deaths due to pulmonary fibrosis were 5.2% higher in the fall, 17% higher in the winter, and 12% higher in the spring months. Although the reasons underlying this relationship between the season and mortality are unknown, lower vitamin D levels might be a contributing factor. In a study that examined patients with systemic sclerosis, an autoimmune condition characterized by extracellular matrix deposition and connective tissue fibrosis, their serum vitamin D was lower than in the healthy controls. In a lab study, 1,25-dihydroxy vitamin D3, the metabolically active form of vitamin D, reduced the fibrotic transformation of the human lung epithelial cells in response to stimulation by transforming growth factor beta. In a mouse model, vitamin D supplementation decreased pulmonary fibrosis induced by bleomycin, a toxic anti-cancer drug. Polyphenols are a large group of plant compounds with powerful free radical scavenging and anti-inflammatory action. Polyphenols include flavonoids and phenolic acids, and they're an important component of the diet, and they've been studied for their many health-promoting and anti-aging properties. Numerous animal and lab studies have examined the potential role of polyphenols in preventing and treating fibrotic lung disease. Many polyphenols have shown promise in reducing fibrosis in animal models of idiopathic pulmonary fibrosis, increasing the antioxidant enzyme activity and/or inhibiting specific inflammatory signaling molecules involved in the progression of idiopathic pulmonary fibrosis. Some of these polyphenols include curcumin from turmeric, quercetin from onions, apples, and other plants, resveratrol from red wine, grapes, and berries, caffeic acid phenethyl from bee propolis, apigenin found in parsley, celery, and other herbs and vegetables, and gallic acid found in tea, the EGCG or epigallocatechin gallate from green tea, citrus flavonoids from citrus fruit, magnolol from magnolia bark, polyphenol-rich extract from pomegranate, and magaferin from mango leaf. And finally, dihydroquercetin from grape leaf, salidroside from Rhodiola rosea, proanthocyanidins from grapeseed, amodin from rhubarb, and salvinolic acid from red sage. Curcumin is a yellow flavonoid and is found in turmeric. Its anti-inflammatory and free radical scavenging properties are well known, and they contribute to its potential benefit in pulmonary diseases, including idiopathic pulmonary fibrosis. A number of preclinical studies have found that curcumin can reduce lung inflammation and oxidative stress, and it can prevent experimentally induced pulmonary fibrosis in animal models of idiopathic pulmonary fibrosis. Animal research also suggests curcumin can prevent fibrotic changes in lung fibroblasts, and in lab studies, it can reduce inflammatory signaling in the cells that line the lungs of the alveoli. Quercetin is a flavonoid, and it's commonly found in fruits and vegetables. It has anti-inflammatory, free radical scavenging, and possibly senolytic properties. In lab research, it was shown to suppress the production of collagen in fibroblasts by acting on a molecule known to be involved in several lung diseases. Orally administered quercetin has been found to raise antioxidant enzyme levels, decrease levels of markers of inflammation, prevent oxidative lung damage, and reduce fibrosis in rats and mice with experimentally induced conditions similar to idiopathic pulmonary fibrosis. In a preliminary study, quercetin reduced the production of inflammatory cytokines when incubated with blood from idiopathic pulmonary fibrosis patients. And interestingly, the anti-inflammatory effect was less evident in blood from healthy controls than in the blood from the idiopathic pulmonary fibrosis patients. Resveratrol is a polyphenol and is best known for its presence in red wine. It's also found in grapes, berries, Japanese knotweed, and peanuts. Like quercetin, it has anti-inflammatory and free radical scavenging capacity, and its demonstrated anti-cancer and cardioprotective potential. Resveratrol has also been shown to inhibit the proliferation and the collagen production by fibroblasts from idiopathic pulmonary fibrosis affected lungs. Resveratrol has been found to protect the lungs against injury and fibrosis in animal models of idiopathic pulmonary fibrosis. In one study, resveratrol lowered lung tissue injury, the fibrosis, and the death rate in mice who were exposed to a fibrotic agent, and it also increased the activation of sirtuin-1, a protein that has an important anti-fibrotic function. Caffeic acid phenyl ester, a flavonoid extracted from bee propolis, has numerous biological activities, including regulating the immune function and reducing inflammation. Caffeic acid phenyl ester was found to prevent collagen deposition in the lungs of animals with an experimentally induced idiopathic pulmonary fibrosis-like condition. It was also found to decrease lung and plasma levels of key pro-fibrotic and inflammatory molecules, and it reduced lung tissue free radicals in the animal model of idiopathic pulmonary fibrosis. Treatment with propolis also inhibited the pro-fibrotic signaling in human alveolar cells triggered by an inflammatory molecule implicated in idiopathic pulmonary fibrosis. Several other types of plant chemicals or phytochemicals have been found to alter inflammatory and oxidative pathways to fibrosis and normalize alveolar cell signaling and fibroblast activity in lab experiments. And it's shown promise in preventing or reversing pulmonary fibrosis in animal models of idiopathic pulmonary fibrosis. Some other phytochemicals are ginsenoside from Asian Panax ginseng, notogenoside from Chinese Panax ginseng, and glycyrrhizic acid from licorice, Schisandrin B, a lignin from Schisandra chinesis, diallyl sulfide and related sulfur compounds from garlic, and sulforaphane from cruciferous vegetables like broccoli and cabbage. For more on herbs, supplements, and natural treatment plans, check out my website.